So, so most of you will be, during your PhD work, you will be dealing with all these methods, and I'm just giving you a brief idea, like those techniques which are commonly used or may be considered as a molecular diagnostic, techniques. And these techniques have various implication in terms of research as well as in terms of diagnosing any sort of, human health or disease conditions. So this molecular diagnosis comes under a big umbrella of healthcare diagnostics. So molecular techniques, again, as I mentioned, that it can be used in research setting as well as in the clinical setting. So most of the things that, because we work in a hospital setting, so most of the things that you will be doing in terms of your PhD work, ultimately you will be either designing some hypothesis that I'm going to develop a biomarker, I'm going to identify certain mechanisms, I will be investigating the role of certain pathway. So the ultimate aim of your thesis would probably be identifying certain biomolecules or certain markers or certain disease mechanisms which can be applicable to the translational research or maybe directly applicable to the clinical settings in terms of improving patient care as well as the human health. So this healthcare diagnostics is one of the part of a healthcare system. So when a patient comes to a clinic, how do a clinician decide whether he's a healthy person or he's a diseased person? So generally, in terms of diagnostics, there are two major approaches which are followed. First is the diagnostics imaging, in which one can identify a disease condition in terms of producing certain images of bodies and organs, in which they can identify whether they are bit different than the normal cases. So CT, that is computer tomography, MRI, magnetic resonance imaging, PET imaging, SPACT, ultrasound. So you might have heard all these things, and this is one of the aspect of diagnostics that we call it as imaging diagnostics. So in this case, mostly we see or we observe the changes in either using radioactivity or maybe some fluorescently labeled tracers that can be incorporated in the body, and then eventually it can be scanned through various high-end equipments, and also based on the sound waves like ultrasound. So these are some of the common methods of diagnostics that are categorized under diagnostic imaging. Similarly, in other categories, so this is without having any sort of taking out any sort of samples from them. But there are, when we are taking any bodily fluid, for example, blood, tissue, sputum, urine, or maybe in today's time we are also dealing with COVID. So the oropharyngeal or nasal swab that is used for detecting the viral presence, virus presence. So those things are categorized in the in vitro diagnostics. So when we take out certain samples and then we try to see various molecules, whether it can be a protein, it can be DNA, RNA, or maybe some metabolites. So that is characterized into in vitro diagnostics. And these in vitro diagnostics can further be classified into analysis of various parameters, and these are from the clinical chemistry point of view: immunological parameters, hematological parameters, pathological findings, microbiology, and eventually the molecular diagnostics. So each one of these having have overlap with each other, and that can be categorized in vitro diagnostics. So molecular diagnostics is a part of in vitro diagnostics, which is again comes under the umbrella of healthcare diagnostics. Okay, so now we will be focusing mainly on the molecular diagnostics part, which involves various molecular biology techniques, and we will be discussing briefly one by one. So what is molecular diagnostics? So when we are dealing with various, so diagnosis doesn't mean that we are always looking for certain disease conditions. It can be also used to identify a person's health, whether they are healthy or not. So nowadays, people go for a routine checkup, and in those cases, sometimes your all vitals and other stuff are okay. So that means if you are healthy, your indicative markers should be fine. But now there are various advanced method which can tell you whether you are predisposed to certain conditions, if you have certain changes in the biomolecules. So overall, uh, a person, or the method of identifying a person's health or disease condition through changes in biomolecules, and these biomolecules could be DNA, RNA, and protein, right? So these changes can be seen either in the cells, which can be isolated from a tissue that through the invasive procedures, or maybe, in the body fluid, for example, urine, or in the blood samples or plasma or sputum, everything. So in those cases, if we are looking for certain biomolecules, DNA, RNA, or protein, and that can be classified under molecular diagnosis. So how do we do that? By using the combination of various molecular biology techniques. So that will be discussed later on. And these techniques will not only tell us about the qualitative analysis, but as well as the quantitation. So whether they are present in how much quantities, whether they are below threshold or above threshold, that can also be used in terms of defining a health condition or defining the extent of healthy or disease condition. So what we basically look at, either the presence of a genome or maybe the proteins which are encoded by those genes. So that genome could be human if we are looking for certain specific type of mutations. So, and then viral genomes, microbial genomes, if there is an infection condition. Okay, like for example, in case of COVID nowadays, we are seeing I think most of you might have gone through this COVID testing, right, before joining, or maybe, in one way or another. So every time, they used to take out certain samples, and then that sample goes to the lab. And I'm wondering if anyone knows about this, like, real-time PCR. I'm sure that most of you are quite aware of real-time PCR, right? But can anyone tell me what is TrueNet, right? You might have heard about TrueNet. So can anyone tell me if you have some idea what is TrueNet? No one? Okay. Okay. So, okay, that's fine. So I'll discuss a little bit when we go further. Okay, so these are the molecular methods that can be used to basically measure the changes in the biomolecules, for example, DNA, RNA, or protein, and they have a greater implication in terms of, previously those these I think the disease condition either visualized by anatomical changes as well as histochemical changes. So generally, there are certain samples which were taken from the blood or maybe cells, and then they were seen microscopically, and if there is any changes in their structure, in their features, then they can be indicative of certain diseases. And then eventually, to confirm that, there are various biochemical tests were performed, and nowadays those biochemical tests were again supplanted with numerous molecular diagnostic procedures, in which we can now confirm various type of diseases, their stages, their prognosis, their subsequent progression by using various methods. So this molecular diagnosis has very greater implications in the various fields, including oncology, infectious diseases, genetics, and clinical chemistry. And it is advantageous in terms of developing personalized medicine. So a person, how they are responding to certain therapies, it can also be identified based on their genetic profiling, their gene expression profiling. So that is a very important aspect of molecular diagnostics. So overall, molecular diagnostics deal with the identification of various biomolecules, specifically DNA, RNA, and proteins, in biological fluid or maybe in the biological samples, through the use of molecular biology techniques. And we use it very frequently. And it is very, again, the implication of this molecular diagnostics is in the risk assessment. For example, if somebody is, predisposed or susceptible to various risk factor, then it can be diagnosed by various tests. So it is not about the molecular diagnosis, but generally the diagnostics, the role of diagnostics in terms of, patient care. What are the clinical implications and what is the description? I'm going to describe it here. That risk assessment is one aspect which will tell us how to avoid those kind of diseases by implementing wellness programs. And there are various methods by which you can screen if some individuals or some population is at high risk. So a screening program also involves various methods of diagnostics, in which for the results of this, that we can identify, or we can at least learn about the disease at the very early stage, so that disease management can become easy. And then diagnosis also used for definitive diagnosis and general typing. So screening, after screening, if we find certain individual which are vulnerable or having symptoms, then it can be diagnosis at maybe at what stage it is there, or maybe which kind of specialist should be seen, it should be sent. So that refers to the appropriate specialist. Staging and prognosis again is one of the important aspect in which with the use of molecular diagnostics or maybe other diagnostic methods, we can identify different stages, recurrence, and how they will be responding to various sort of therapy. So it also helps to determine what kind of therapy is required. And then again, therapy selection, and also with the help of treatment, we can also monitor how the disease, whether disease is progressing or it is, having some sort of improvement. So this is the overall general role of diagnostics. And if we talk about the molecular diagnostics, I can give you some of the examples in which cervical cancer screening. So cervical cancer is second most common type of cancer, in India. Still, previously it was number one. Now it has been replaced by breast cancer. So cervical cancer screening programs are very common. Our department also used to held these programs every year, and in which, so conventionally what people used to have this pap smear test, in which there are it's a kind of histo cytological analysis in which there are various type of cells which are abnormal, and based on that people suggest that there may be some sort of infection in the cervix. So that is now replaced by HPV genotyping, in which we can surely say that whether this belongs to the cervical cancer or HPV viral infection, and what kind of infection it is, like it is high risk infection or low risk infection, and even in the high risk, what kind of genotype it is. So that helps in the screening as well as developing, proper disease management. Similarly, in terms of identifying various gene mutations or genetic disorders, cystic fibrosis is one of the example in which a particular gene CFTR is mutated, and that can also be checked before birth as well as after birth. So amniocentesis is one of the method in which certain fluids were taken from the pregnant woman, and then it can be checked whether the newborn which is coming up are having if the genetic profiling is okay or it has certain mutations or not. So these kind of things can be used. And nowadays, due to the COVID-19 infection, we know a lot of about RT-qPCR. So RT-qPCR stands for reverse transcribed quantitative PCR. So real-time PCR, qPCR, the similar term, but this RT means reverse transcription. So there are various types of tests available nowadays for identifying COVID-19 infection, and that also comes under the umbrella of molecular diagnosis because we deals with the real-time PCR here. So that's why we can classify them in the molecular diagnosis, and it is more sensitive than the antigen antibody test. So you also seen various ELISA-based methods now coming up that can tell you about whether you are infected with it or not. But if it is very early stages when the body are still making antibody against the antigen which is not detectable in the serum, that can also be quantified by the viral infection by the use of qPCR. So it can tell us about if there is a very minimum infection of that, or as well as there is a high viral load. So RT-qPCR is a method which is now very commonly used for COVID-19 infection. So these are some of the examples, like with the use of molecular methods, how we can screen, how we can diagnose a disease, how we can monitor a disease progression. And in addition to that, there are various examples in which these molecular tests can also help in taking decision in terms of deciding therapies, that what kind of therapy should be given to the patient. For example, in case of pancreatic cancer, colon cancer, or lung cancer, there are specific therapies which are targeted to EGFR receptors, because in these metastatic cancers, so if there is a metastatic case, in those metastatic cases, EGFR receptor is upregulated. So there are anti-EGFR therapies available to those. But in certain cases during metastatic condition, if KRAS mutations occur, in those cases these anti-EGFR therapy doesn't work. So in those cases, alternative therapies has to be decided. So that helps again to decide what kind of therapy. So this is to look at the KRAS mutation in these particular type of cancer and decide that if there is a mutation, they will not be responding to the anti-EGFR therapies. So if we can find the KRAS mutation in those cancers, we can again alternatively decide what kind of therapy needs to be given. So all these things help us to at least, in terms of deciding what kind of therapy, what kind of disease, what kind of stages that we are in. So altogether, diagnostics, or especially the molecular diagnostics, is one of the important aspects in medical sciences as well in research that will help us to understand. So when it comes to the techniques, again, because we always are more interested in looking at what kind of methods, and I'm sure most of you are very much familiar with this, and you might have studied in your masters or in the previous, work area. So basis of molecular diagnostics again is broadly, it can be classified in various ways, but the easiest way of, to me it appears that we can classify it in different ways. One of the way is to based on their techniques. So one is the amplification techniques in which we have PCR based methods. So PCR, normal polymerase chain reaction, and then if this polymerase chain reaction, generally when we say PCR, we always have this thermal cycler in which we have primers, we have template DNA, we have DNTPs, and that amplifies it, and then eventually you load onto a agarose gel and then see the images along with the markers that you load in the lanes. And then you can qualitatively analyze whether your PCR product is there or not, or maybe what kind of changes in the PCR product intensity. So this is generally normal routine PCR. When you can quantitate it by using a specific instrument that we call as a real-time PCR, or whatever the amount of the product that you are getting is called as, it can be measured by quantitative PCR. And then, so this can be applied to the DNA, right? And when you are dealing with the expression profiling, then you have to convert first RNA to cDNA, and then this cDNA can be used as a template, and that again can be quantitated, like how many copies of RNA is present can be reflected in terms of cDNA, and that can be amplified by using reverse transcribed qPCR. So amplification technique is also one of the major areas of molecular diagnostics in which we detect several gene mutation, SNPs, or maybe gene expression profiling of various genes, which can give us an idea about disease condition or a healthy condition. The second aspect is blotting technique, in which we first take these DNA, RNA, and protein and transfer to a different location, not location, but DNA, RNA, or protein can be transferred to a semi-solid or solid support in the form of membranes or adhesive matrix, and then eventually you detect it by using specific antibodies or specific probes. So southern blotting, dot blotting, these are or RFLP, that is restriction fragment length polymorphism, northern blotting for RNA, western blotting for protein detection. So basically, you are transferring your molecule to a solid support of a matrix and then fixing it, and then eventually identifying with a known probe whether it is present or not. So there are these are the classical techniques nowadays in terms of in terms of methods. Only Western is more popular. Southern and dot blots are rarely used. Sometime RFLP is again one of the method. Northern blotting has been replaced by real-time PCR or maybe qPCR, in which we can measure easily the gene expression profiling. Similar to blotting technique, there are other methods, hybridization technique, in which again, the specific probe is hybridized to either chromosomes or DNA or maybe a small fragment of DNA. So there are various methods which is generally used in molecular diagnostics, are the FISH, fluorescence in situ hybridization, and I will discuss a little bit in detail in later sections. And FISH is a method which is known to hybridize with the DNA fragment, and it also reflect the chromosomes. So if there is a chromosomal aberrations, if there is a chromosomal translocation, if there is any defect in the chromosomal pattern, which can lead to genetic disorders or maybe syndromes, genetic syndromes, that can be identified classically as a karyotyping, and now in a more advanced form we use it as a FISH, fluorescence in situ hybridization. Similarly, so this method is the conventional method or modification of conventional method. Chip or microarray is a more advanced method in which high throughput identification of various gene sequences, whether it can be used for sequencing purposes or it can also be used for detecting gene expression profiling. So these are the examples of hybridization technique. Other methods can involve ELISA, which is very common. I think everybody knows that it is the antigen antibody interaction. and mainly used for protein or antigen detection. There are other methods, flow cytometry, mass spectrometry. So flow cytometry again is based on your fluorescent properties, based on the cell size, shape, and their complexity, and also it can be modified by the use of various specific identification molecules such as cell surface receptors. So that can be used in terms of defining a certain population of cells. So we will be discussing a little bit more. Mass spectrometry again can be used for determining protein or nucleic acids. And NGS is one of the most advanced method that can give a lot of information about the genome and the gene expression as well as various changes in the genome altogether. So if I have to combine all these things, I can give you a good example of the SARS COVID-19. That is the now the very hot nowadays, that everybody is working on this COVID-19 in one way or another. And I think when we talk about molecular diagnostics, it is having everything, so you have this is a typical, just a representation of SARS-CoV-2, and it has various nucleocapsid, spike protein, envelope protein, membrane protein. So these are various, indicators that can be used by using various molecular diagnostic technique, either to detect RNA, for example, by sequencing method, by RT-PCR, and there are various advancements in nowadays in the methods by which you can detect or maybe identify the viral, presence of viral genome, mainly the RNA. And it can also be used, these various proteins can also be used to develop immunoassays, or, can be used with the help of mass spectrometry to identify whether they have the presence of this virus or not. So overall, molecular diagnosis, again, a very important area of research as well as in the clinical system, because ultimately these all things have been developed through research, and there are so many advancements in those areas. So we will be going one by one that what basically these techniques are. So first, coming to the amplification technique, and I'm giving you very relevant example nowadays because you are more familiar with the COVID. So I'm trying to correlate these techniques with the COVID, so you can have a good idea about how these COVID test, or maybe how these molecular diagnostics can be helpful. So quantitative PCR, again, it's a kind of real-time PCR in which you detect exact copies or exact expression of your gene of interest. So, for example, in case of COVID, generally the samples are collected, oropharyngeal or nasopharyngeal samples, and these are transported in the VTM, that is viral transport media. So there's a sample collection center, and then what basically in these viral transport media, there is a system. Either you can just load onto the cartridge, which is automated, and then it can ultimately gives you the purified RNA, or maybe by manual methods you can isolate the RNA. Now this RNA is used for making first strand cDNA, so that is called as reverse transcription. So you need primer as well as you need reverse transcriptase, so you can make single stranded cDNA, and the remaining, sorry, first a single strand cDNA and then double stranded cDNA. So altogether you use this cDNA as a template for detecting. So if suppose n number of copies of viral RNA is present, it will convert to the cDNA, and this cDNA can be used as a template. So you need the primers and everything, and then again the normal PCR happens the same way the real-time PCR happens. The only difference is that because we use normally the probes, which are maybe certain nucleotide long, 70 to 100 nucleotide long, and they are very specific to a particular portion of a genome. In this case, in case of COVID-19, it can be designed either through certain genes, for example, spike protein or nucleocapsid, or more importantly, the most authentic one is the RDRP, that is RNA dependent DNA polymerase. So these are the specific indicators genes that can be used as a probe. So in the real time PCR, you need primers. Along with that, you also need probe. So this probe is very specific to a particular gene. So here what we are doing is basically trying to identify or trying to quantitate the expression of a certain gene. So the probe is added, which is very specific to a gene, and now the primers will attach, and they will be, amplifying the whole fragment. And during the amplification, the dyes which are attached to it, it will release. And the more the dye will release, the more, fluorescence you will achieve, and that will show you that will show us about the threshold level. So more quenching, more fluorophore is released by the DNA polymerase cleavage, and the more fluorescence will get. So more template, uh, then the increase in the fluorescence will be more, and that will come as a threshold cycle. So that is again decided by the CT value. So this is normal amplification. This is another amplification. So if there is a more number of cells, more number of DNA or RNA is present, then the threshold will come sooner. So maybe the CT value will be less. If less number of DNA copies or RNA will be present, then the shift will be, or the threshold will come a little bit later. So CT value again reflects copies per reaction, or maybe the CT value reflect the threshold number of the copies which is present in a which actually tells you about the how many number of copies are present. So CT value if less, then maybe the more number of copies are present. If CT value are higher, then less copies will be reflected. So quantitative PCR generally is a kind of modification of polymerase chain reaction. The difference is, in this case, we generally use a probe which is fluorescently labeled and very specific to a particular gene, and release of these fluorescence fluorophores which are attached to their surface, their ends, can be detected by the real-time PCR detector, and that translate into the threshold cycles. So more number of transcript, less CT value, and these can be used in the various type of modifications for detecting pathogen infection. So this is an example of COVID-19 infection and how we detect gene mutations. Again, if there is any mutation in the gene, whether it will overexpress, whether it will down regulate, specific probes can be designed to find such changes. Similarly, single nucleotide polymorphism or variable number of tandem repeats can also be identified, or can also be quantitated exactly by using these quantitative PCR method. So this is one example of the amplification technique. So PCR, quantitative PCR, and the RTQ PCR, which is reverse transcribed quantitative PCR. So altogether, there is an amplification of the template, and template can be DNA or RNA, but again RNA has to be converted into the cDNA, and then cDNA can be uh, cDNA is a reflective of RNA load, and that can be measured quantitatively. So coming on to the blotting technique again, so blotting in which either DNA or RNA is transferred to a solid support or matrix or membrane, generally we call it as, and then complementary sequence can be used as a radio label. So in case of real-time PCR or quantitative PCR, we were designing a probe which was complementary to the RNA or cDNA. In this case again, in southern blotting, these probe will be generally in case of southern blotting. Initially, people use radio-labeled DNA probes. So they are radio-labeled part of small fragment of DNA which is complementary to the target DNA. So that was so first DNA is attached to a matrix which can be a membrane, and then it is hybridized with a known sequence of a DNA by using radio-labeled DNA probe. And once if it is attached, auto radiography or X-ray scanning, we can find whether the presence or absence of those particular fragment. So that is used generally to determine copy number or any sort of DNA changes. Similarly, Nathan blotting, so here the template is your DNA, here the template is your RNA. Otherwise, the procedure of hybridization and autoradiography are the same. So Nathan blotting actually tells you about the gene expression profiling. Southern blotting, most probably the the copies of how many copies of gene is present in a genome, and the location on the chromosomal wise can be obtained. So northern and southern blotting are again genomic hybridization methods, in which southern blotting deals with the DNA hybridization, and northern blotting with the RNA hybridization. And again, both of these things can be measured autoradiographically with the use of radio-labeled probe. Nowadays, there is an advancement. So now people use fluorescent probes rather than using radio-labeled probe. And third one is the Western blotting. So Western blotting is more common, in which proteins are attached to the PVDF membrane. So for DNA and RNA, people prefer nylon membrane or nitrocellulose membrane, depending on the length or stripping sort of requirement. For protein, mostly PVDF membranes are used. Nylon membranes are also used. Nitrocellulose are also used. So depending on how many times you have to use it, uh, people use accordingly in the various membranes. So proteins are attached to the surface. That surface can be your membrane, and then these are hybridized with the primary antibodies, and then primary antibodies and then secondary antibody which are labeled with that. So with the chemiluminescence, you can detect the presence or absence of any protein band or maybe the expression of that. So these are some of the blotting techniques. And now, uh, like I mentioned before, the radio labeled probes are now replaced by fluorescence probes and multiplexing for identifying more than one target. Okay, so this is the blotting technique. Now coming on to the third part, which is your hybridization technique. Um, um, so in hybridization, one of the major area is the fluorescence in situ hybridization, which is basically to detect. So in this case again, it's similar to southern hybridization, but in this case you are hybridizing directly with the chromosomes. So probe or DNA can be designed and it is labeled with the various fluorescent molecules, and then it is denatured it is denatured, and at the same time target DNA, which is either in the form of metaphasic plate or various methods, it is fixed, the chromosomes are fixed, and then you can hybridize it with the known labeled probe, and then it can be visualized later on. So this fluorescence in situ hybridization is primarily to detect, changes in the chromosome numbers or, chromosomal ploidy, aneuploidy, euploidy, detection of various microdeletions, translocations. If there is any aberrant gene expression of certain genes, that can also be visualized, mapping of newly isolated genes, identification of various, genetic disorders. So these are the this is one of the method of identification of those particular changes, and this is one of the example of karyotyping which are used in general to see the pattern or chromosomal pattern. If there is a deviation from the chromosomal standard pattern, then and specific chromosomal changes can be identified, and it is reflective of various disorders like Down syndrome, Fragile X syndrome. There are so many genetic disorders which can also be characterized by changes in the chromosomal pattern. So each and every chromosome has a banding pattern, and if it differs from the original one, then there is an indication that there may be some genetic abnormalities. Similarly, one of the example is translocation. Ewing sarcoma, there is certain rearrangements in the chromosomal part, and so this is the normal case, and there are certain chromosomal portion which is translocated, and that is considered as abnormal. So this is basically to identify those particular features by using the hybridization technique, and this is these are the examples of your fluorescence in situ hybridization. So this fish is generally a method. Again, it is similar to western blotting, sorry, southern blotting, but in this case we are using chromosomes for hybridization, and the probe or the target molecule will be hybridized or would be hybridized with the probes that are specific to a particular gene or particular chromosome belonging to a particular chromosomes. So by that way we can have idea about the chromosomal differences, chromosomal aberrations, or maybe changes in the banding pattern. So these are the hybridization. So this is one of the method of hybridization. Another hybridization method is more advanced, that is called as microarray. And again, it is modification of your southern blotting. So in southern blotting, um, this DNA is attached to a matrix or maybe the membrane, and then you hybridize with the probe. In this case, um, the probes are designed and they are already been pre-coated on certain plates, which are multiple, having multi very fine um size of milli or micromolar micrometer range, and these products or PCR products are adhere, or you can say attached to the surface, and eventually these probes, because they are already known that how many copies of these probes are attached, and they are hybridized. So for example, if one has to identify how much sample one is differing in terms of expression of RNA with the sample two. So in sample one, they isolate total RNA and then they label it certain dyes, and that will be specific to the sample one. And the sample two, again, it is labeled with a different dye. So if sample one and the probes are known sequences which are representing particular RNA, and also how many copies are there, that is also already known in this case. So you add the sample one and sample two, maybe together or maybe in different ways, and then attached to that, you can see the detector or image analysis. So based on the intensity of the respective dyes which they are labeled with, you can easily see the raw images, and then later on these images are analyzed in terms of differentiating this expression with this. So green versus red, here clearly see how many are giving signals, and the intensity again tells us about expression level. So these microarray can be used. These are the basically contain thousands of immobilized DNA sequences, and then DNA sequence either can be used for sequencing purposes or can be used for gene expression analysis. So these DNA sequence, if we know already how many copies of these immobilized DNA sequence are present, and then based on the attachment of various sample molecules and the intensity and their respective intensity, can tell us about how many copies are present. So this is one of the way of looking at gene expression profiling as well as sequencing. So microarray again is also one of the method of sequencing, although it is not very much used nowadays for sequencing. But for gene expression, still people use microarray data as one of the authenticated, method. And this DNA microarray also has a very diagnostic applications that, like I mentioned, that it can also be used to diagnose, disease diagnosis through biomarkers. So genotyping, determination of disease-related genes. For example, basically it is about gene expression profiling, post-translational modification screening, single nucleotide polymorphism screening also it can be used. So it can be used to, by using various biomarkers, which can be, identified through research or, biomarker-based research-oriented proposals. So that can be identified. Genetic disorders can also be identified. For example, chromosomal abnormality analysis, mutation analysis, SNP detection, and also presence or absence of various infectious agents such as bacteria, virus, or fungi. So altogether, hybridization techniques, primarily the microarray, can be used for various applications, and genetic disorders and infectious diseases are very commonly used methods for detecting all those changes. So this is second aspect, hybridization technique. The other methods, so far what we have seen, primarily the amplification technique, blotting technique, sorry, blotting technique and hybridization technique. So in hybridization, fish and chip or microarray. Now there are other methods which everybody knows. I think most of them are quite familiar with ELISA and flow cytometry, and I will be a little bit more discussing about mass spectrometry or next generation sequencing. So there is no need to worry. You will be discussing all these methods in coming classes. And if someone any question, if they want to understand in more detail, you are most welcome to discuss this thing over the phone or maybe, through the, video conferencing. I'm always available. So some of the techniques will be discussed eventually in the same, foundation course in detail. For example, Western blotting, fish, or maybe ELISA and other methods. So there is no need to worry. I'm just giving you a brief overview in terms of its application in molecular diagnostic laboratory. So far we have seen amplification, blotting technique, and hybridization. Now we will be looking mainly on more advanced methods apart from ELISA. ELISA is very common and very well-known method. The flow cytometry, mass spectrometry, and NGS. So, so if we look at, oops. So So just to wrap up with this ELISA method, ELISA, everyone knows that ELISA is again immunological enzyme-linked immunosorbent assay. So in which the primary goal is to identify antigen, and this antigen can be a viral particle or it can be certain protein molecule. So this antigen can be attached to a solid surface. Solid surface means it is attached or pre-coated in a 96 well plate format, or maybe it is attached to certain membrane. And then these molecules are identified by adding their specific antibodies that we call as primary antibody. So if the antibody is already linked with an enzyme, which is, which can react with a substrate, so that is what we call it direct ELISA. So direct ELISA directly identify antigen without the addition of any secondary antibody. So antigen is a virus particle or maybe certain proteins that we want to identify, and this antibody is again complementary to this antigen. So this is the antigen antibody interaction, and if we have detecting substrate or detecting enzyme attached itself to the primary antibody, that is called as direct ELISA. So when this primary antibody does not have enzyme linked to it, but we require additional antibody or secondary antibody for detecting this primary antibody, then it is called as indirect ELISA. And sometimes to increase the specificity, we use sandwich ELISA, in which the antibodies which are specific to certain antigens are first coated on a plate, and then your samples in which you have your antigen can be added, and then this antigen again is bound to next secondary antibody, sorry, the primary antibody, and then this primary antibody is again bound to the secondary antibody which has enzyme linked to it. So sandwich ELISA is slightly, a little bit complicated, but in direct or indirect ELISA, you can directly identify antigen which are attached to the surface. So these are basically the principle of antigen-antibody interaction, and it is a typical diagram or typical images of ELISA plate in which you see yellow color showing, in some of the cases the yellow color is too dark, or sorry, too intense. In some of the cases, maybe the yellow color is less intense. So based on their intensity, you can also quantitate exactly how many or antigen or how much your target is present. So this is basically, nowadays there are so many kits are available. In fact, COVID also, for COVID there are various ELISA kit available. So that can detect IgG or IgM type of antibodies. So IgG, I think primarily, IgG is the first line responder, and IgM is the second, sorry, the more specific one. No, IgM is the first responder, and then IgG will retain for a longer period of time as a memory. So ELISA is one of the method, or it can also be classified in molecular methods in which we specifically detect proteins. So previous methods are based on the DNA, RNA methods, and this is one of the method for the protein identification. Um, there is another method, flow cytometry, which is more commonly used in terms of detecting hematological malignancies. So this flow cytometry will be discussed in detail in, I think, maybe the later section or maybe the last class of this course in detail by Dr. Rahman, because he is an expert and he deals with the flow cytometric facility in our institute, and also he is an expert in the detecting various hematological disorders. So just to give you a brief idea what we can do with the flow cytometry. So again, I am giving you perspective in terms of their use in clinical applications, but they are very well used in the research applications also, and they are very much they have a very great implication in terms of understanding our research questions. So whatever planning you are doing for your research work, you can use these methods to actually improve your existing knowledge, as well as to incorporate new ideas to achieving your target. So flow cytometry also is one of the method which detects certain type of cells for measuring their area and shape, their intensity, their texture, their correlation, their granularity. Okay, so this is just a basic features of flow cytometry, in which cells are passed through a chamber and they are made a single cell suspension, and when they pass through this very fine uh, chamber in which only one cell at a time can pass, the light source will tell us about their area and shape, their intensity, texture, and if those cells are labeled with certain sort of probe, for example fluorochrome, which are attached to the antibody, and if this cell may be a healthy cell or maybe a defective cell, or maybe a cancer cells which are expressing more certain type of receptors. So those cells can be marked with certain antibodies, either CD receptors or maybe various types of receptors. And then based on the fluorescence of these fluorochromes, it can be classified or it can be, identified that in a given sample, even given tissue samples, or maybe in a given cell suspension, how many number of cells or how much number of cells are expressing that particular type of, receptor, because that receptor can be identified by antibody, and this antibody is attached to a fluorochrome, and this fluorochrome can be identified with the laser light which are specific to a particular wavelength. So one cell at a time having a specific tag on it, and that can be detected. So this is basically flow cytometry based on their fluorescence properties. If we just want to measure in other properties also, like area, intensity, or their complexity, that can also measure. So this is one of the example of in these cases. For this is this is the analysis of a leukemic patient in which 20% of the cells have some deformity in their structure and 80% cells are normal. So in this case, now, since it has 20% of the leukemia cells, a personalized target can be developed, that because still it is in the early stages. So maybe in those cases, treatment strategy can be different. So flow cytometry can be used for identifying various properties of cells, and also can be identified or also can be used to identify various type of cells depending on their surface properties. So generally, flow cytometry is used for the diagnosis of hematological disorders. Now coming on to the more advanced methods. So flow cytometry and ELISA, they are used. And in addition to that, there are now upcoming areas in which people are now using more and more advanced methods. Although these methods, again, when they are used in isolation, their cost is more, they are very expensive tests. But when there is a bulk of samples, they will be more cost effective because they are highly, accurate, and their speed is quite high. They are high throughput analysis. So always their reproducibility is very good, and but only the main problem is that their prices are very high. So it is more expensive. But again, if the sample size is good enough, it can be, very cost effective. So mass spectrometry is generally used to detect nucleic acid as well as proteins, and they are basically identified. So it's a separation technique primarily. It is very rapid, cost effective, and highly reproducible technique. It also it identify molecule based on their inherent properties of mass to charge ratio. So mass, molecular mass, and charge. So these molecules, or these, either it can be a protein molecule or it can be a DNA or RNA. First, they are charged with some sort of ionization, and for ionization, there are various platforms such as MALDI or ESI. So metric associated laser desorption ionization, and that is electron spray ionization. So these are two major platforms that are used for mass spectrometry. In ESI, generally, these nucleic acids or proteins are dissolved in organic solvent, and these organic solvents are applied again to a to for ionization. So if so, these molecules are sprayed in ionization chamber that makes them ionized, and based on their ionization and the movement, their mass to charge ratio decides what is the size, how they are traveling to the vacuum. So basically, if you look at this, so this is identifying nucleic acid or protein. So these are the process which are done outside the the MS mass spectrometry instrument. So sample, you prepare, you take the samples, isolate the nucleic acid, and then do the multiplex amplification like the normal PCR. But although, sorry, normal PCR, it's a multiplex amplification in which you have a probe or a specific fluorescently labeled DNTPs, which makes the overall reaction quite fluorescent. And then you isolate, or then you purify all those fragments, and individual fragments then go to the a particular sort of setup in which they are first ionized, and then it moves to a certain vacuum chambers, and based on their M to Z ratio, they are identified, and this is attached with the detector which can detect their mass to charge ratio as well as fluorescence, and based on that they generate the report basically telling what kind of protein or how much, sorry, what sequence basically or what size of the nucleotide is there. So that can be detected by ESI-MS, and it is again very efficient. So ESI-MS is generally used to detect smaller fragments of DNA, RNA, and proteins. And the more advanced and more commonly used is the method is multi-TOF, time of flight. So time of flight is basically again based on the mass to charge ratio. The difference between these two methods, that this is based on the ionization spray and this is matrix associated laser desorption assay. So in this case, there are various ways, hot and cold, in which not the small molecules, but also bigger molecules can also be identified. So in this multiplex reaction or various combinations of probes can be used to identify different types of SNPs altogether. So you multiplex reaction and then there is a, so these are the two different methods, one in which one is identifying SNPs and second one is just to see the expression of genes. So multiplex amplification, SNP extension, and then desalting. In this case, you prepare cDNA and then you amplify it, and then the remaining product is desalted. Means basically is a purification. And now after purification, each and individual fragment is passed through this MS system mass spectrometry, in which they are basically separated based on the mass to charge ratio. So altogether, MALDI-TOF or ESI-MS, they are very useful in terms of detecting a viral genome or microbial genome as well as proteins, and also can routinely be used for detection of specific mutations which can be useful for therapeutic improvement or various types of find genetic relatedness among viral variants. So MS is again a coming up area, but hopefully nowadays it is most frequently used in nucleic acid and protein identification method, specifically for microbial and viral infections. Although it is slightly expensive, but again, depending on the sample size, it can be scalable and can be made cost effective. The another one is the most important and very much talked method is the NGS, that is next generation sequencing platform, and they are very useful. So if we go back to the history, uh, Human Genome Project was, I think, started in 1990, and it went, around 12, 13 years. So almost, after 12 or 13 years of sequencing the whole genome, human genome was sequenced around, it took around 13 years, and it took around almost $3 billion. So now, if we compare that time with this time, I think you can get your sequence within a week or maybe less than a week, and it will cost you around only $1,000 or maybe less than that. So there is a big difference now and then, because that was the first time. So almost 3 billion, 3 billion nucleotides were sequenced. Almost a similar amount was spent, and it took around 13 to 15 years to finally get annotated sequence altogether. And this is now very much reduced in terms of cost as well as in terms of timing, and also the reproducibility or effectiveness. So this is only being happening because of the advancement in the sequencing technology. And this is one of the example of next generation sequencing, a platform which are used on both of the type of nucleic acid for RNA and DNA. So NGS is this at the moment. NGS is primarily used for DNA and RNA and its modifications. So there are various applications in which DNA sequencing, RNA sequencing, targeted DNA profiling, or long non-coding RNA sequences, microRNA, riboseq, single cell. So there are various sort of applications of these NGS which are generally used in terms of research, and some of the applications are again they are used as a in clinic diagnostics, or maybe genetic testing, or maybe giving you more genetic information on the disease perspective or healthy conditions. So if we look at the NGS, originally when human genome was sequenced, first time the Sanger sequencing, which is again known as a gold standard, and it was used. And during this process, what would have happened, the DNA fragmentation. So for Sanger sequencing, this is just a flow chart of how the whole genome was sequenced. So if there is a DNA, you need to fragment it, and then each fragment needs to clone. and then these clones can be screened in generally like blue-white selection or bacterial. If you are cloning in a certain bacterial cloning vectors, then you will ultimately identify certain colonies which are positive or negative. If they are positive, then you take out those positive clones and then prepare the plasmids, and then with the help of known plasmid sequences, you can identify those particular um, unknown sequences. So for the Sanger sequencing, you require primer, you require template, and then addition of DD NTPs will tell you where the reaction is terminated. And then when you can club them together or analyze the data, then you can get the whole sequences. Although the Sanger sequencing again is limited to 600 to 700 base pair at a time. However, this is reduced, or this is basically improved by NGS system, in which the major fragmentation part is the same. But from fragmentation onwards, now you have various other methods which can improve the outcome of the reaction. And instead of cloning individual fragment, now they are ligating to the adapter, and then this each adapter is already been labeled with some sort of probe, and also their primers are also known, and these adapter ligated fragments are now is used as a bridge PCR or emulsion PCR, and ultimately they are amplified. Individual fragment is amplified multiple thousands or million times, and each time their amplification is recorded with the help of various fluorescently tagged nucleotides. So NGS again is very important platform, which is much more efficient, much more robust, and it can give you multiple, or it can give you it is having more applications than any other method. So there are the various commercial vendors which can provide various NGS platforms, and every individual platform has its own advantages and own disadvantages. But overall, if we talk about NGS and compare it with the Sanger sequencing, it has several advantages, that there is no in vivo cloning, transformation, colony picking, and you just need the adapter ligation. So there is a various, there is a complete set of workflow that requires for this NGS, and there are various quality control mechanisms that are important in terms of NGS. So those methods can be used. If we dig down further, so if we can, if we want to slightly look more closely about the NGS, the library preparation, clonal amplification, and cyclic array sequencing, these three are the major steps of NGS, in which one has to first prepare the library. So library means after DNA fragmentation, you attach the adapter. Now the adapters are attached because adapters are the known sequence of DNA that are already known, and now they are attached to unknown sequence of DNA. And based on these known sequences, you can have the primers to amplify the unknown part. And as soon as you amplify the unknown part, you are detecting it with certain methods. And what are those methods? Either through making them emulsion PCR. So first is making DNA fragments and adapter ligation, and these adapted ligated molecules can be put them in a sort of bubble-like structure or emulsion, oil emulsion PCR, in which individual fragment goes to a specific emulsion, and then and this emulsion is getting amplified every time, and each time when it is amplified, we can detect it by various methods. Or alternatively, it can attach to a surface which is already having known sequences of those adapter molecules, and the complementary strand will bind, and again it will attach to, it will make a bridge like structure, and during that it will amplify again. So there are various methods or various technologies, but overall if we try to based on various methods, if we want to classify them, there are three major techniques that are used for NGS. One is the pyro sequencing method, which is again outdated method. It was very well known before 2008. Now it is obsolete, so nobody is using it right now. But again, just for your information, the pyro sequencing. So once you have DNA fragmentation and adapter ligation, followed by emulsion PCR. So this during the emulsion PCR, what happens that you detect that the method of detection is basically uh, is that DNA polymerase, when there is an addition of any nucleotide, there is a release of pyrophosphate. So phosphodiester bond, when it is formed, 5 prime hydroxyl, 5 prime phosphate group with the 3 prime hydroxyl, there is a release of pyrophosphate. And then if there is a release of pyrophosphate, it is recognized by, or it is ultimately used by an additional assays, which is an ATP sulfhydrylase. So this pyrophosphate is again used by an additional enzyme, and then that makes ATP, and then this ATP is again utilized by luciferase and releases light. So every time if there is any addition of nucleotide, it is detected by the release of pyrophosphate, which is then utilized by another enzyme system and give rise to a small peak of that. So that is one aspect of 4-5 so sequencing or pyrosequencing. Similarly, there is another method which is called a solid platform, in which sequencing by ligation. So here it is a DNA polymerase act. There is another method in which DNA ligase is added. So in this case, 2 to 8 nucleotide probe are added. So this again, it is just to have some idea, although the individual method is very deep and very much detailed. So for that, we have to take individual classes. But again, just to give you a brief idea, that this method is based on release of pyrophosphate. This method is based on the ligation to the probe, and then each probe is specific, and based on the ligation and release of that, it is identified by the detector. The third one is the most common one, which is sequencing by synthesis. So which is more or less similar to the dideoxy method. The only difference is in the dideoxy method, chain terminates when there is an addition of DD NTPs. Now in this case, it is not DD NTPs, but the DNTPs are modified in their 3' end. So they have a blocked 3' end. So only when you deblock them, then there is further addition of additional nucleotide. So this is sequencing by, so individually different fluorescent labeled ATGCs are sent in a small flow, and then if there is a complementary nucleotide is available, the remaining will flush out, and the attached one will be detected by their specific fluorescence. So all these nucleotides are attached, they are labeled with different fluorescent fluorochromes, and then specific fluorochromes representing specific nucleotide will be used to determine the sequence. So this is sequencing by synthesis. So mostly in today's time, most of the very commonly used, whether it's a Illumina sequencing platform or maybe the ABI sequencing platform, they are mainly based on the sequencing by synthesis method. So this is NGS, and again, running an NGS is not easy, because before starting NGS method, or after getting data, it is always difficult to analyze, because it is hell lot of data, and it depends again what kind of sample you are dealing with, and how much reliability of data that you require. So depending on the amount of data, you can determine various aspects, but overall, if we look at the NGS application, it is huge. It can be used for whole genome sequencing. It can be used to identify regulation of various genes for a particular disease. Epigenetic changes, for example, DNA methylation, acetylation, can also be identified by these NGS methods. Metagenomics, paleogenomics, transcriptome analysis, resequencing. These are some of the important applications of NGS, which are very useful in terms of understanding disease perspective. Okay, so NGS. So if we have to look at the overall molecular diagnostics, molecular diagnostic can be an important tool to identify infectious diseases, and COVID is one of the best example nowadays, and it is very relevant. You can clearly identify with use of these various techniques that I mentioned. I think most of the research community nowadays are using these methods to learn more about the COVID-19 or COVID-19 virus infection and associated problems. So in general, molecular diagnostics are used for detecting infectious disease agents, that such as viruses, bacteria, parasites, or fungi. It can also be used to detect novel infections, for example H1N1, COVID, Zika, so on and so forth. Um, it is has a greater implication in terms of identifying various genetic disorders, syndromes like sickle cell anemia, Fragile X syndrome. So these are either single nucleotide mutations or maybe translocation or maybe differences in their chromosomal bending pattern. These methods can also be used for entity determination. So there is now a separate sort of various type of agencies can detect, for example, in case of forensics, transplantation, parental testing, twin zygosity. These are some of the important aspects which can determine the hereditary or maybe the lineages, uh, whether it is related to disease or in terms of identity. So this is just to sum up everything, that in a typical oncology lab, if we say that how the molecular diagnostics is relevant, so it has a very different dimension. For example, in case of hereditary cancer, so we know that hereditary cancers are those cancers which occurs through family history, and some of them are more dominant sort of nature, and some of them are, even though you carry a mutation, but if it is not in both the alleles, maybe it is not symptomatic. But yes, in some of the cases they are very much predisposed. So hereditary cancers or syndromes can be detected by using various molecular techniques, and those assays are maybe recurrent mutations by using PCR, single gene analysis, multi-gene panels. So nowadays, if we, because oncology, in case of oncology, this molecular diagnostics play a very important role, not only diagnosis the cancer, but also kind of deciding the therapies. So, and also for screening purposes. So if in a family, there is a history of cancer, and if somebody wants to know whether he or she is predisposed to certain type of condition, they can always go for molecular diagnostics, in which these are the methods that can be used for identifying hereditary cancer syndrome, whole exome sequencing, and also in the cancer patients. So this is the normal case to detect the hereditary cancer, but in the cancer patients also it can tell you whether there is a chances of having second malignancy or subsequent malignancy. So based on that, choice of treatment can also be decided. And healthy people can also be used, can also identify certain predisposed, or they can be, if they are predisposed to certain conditions. I think these methods can also help them to find their vulnerability or susceptibility to certain conditions. Okay. And then predictive markers again. So this is related to genetics. We can also have a panel of predictive markers that can be used to identify a particular type of tumor or cancer. Assays can be based on DNA, RNA, proteins, or various type of histopathological examinations. Molecular targets can also be can also be identified, means whether they are mutated first, or whether their gene expression profiling are different, or maybe their protein expression profiling are different. So all these things can be measured if we know the predictive marker. So if there is one mutation, but then there is subsequent chances of having more mutations, or subsequent chances of altering various signaling pathways, can also be identified by these molecular methods. Tumor phenotype, for example, microsatellite instability, assays. This is generally used in case of colorectal cancer. BRCAness, again, breast cancer, and mutation burden. So all these assays or all these methods are used as a predictive marker. And to analyze these predictive markers, these different types of assays are used, which are dependent on various biomolecules. In addition to that, circulating tumor fragment can also be used as a molecular diagnostics, in which one used to identify circulating tumor cells in the circulation. So there is a tumor, you release certain tumor cells in the circulation, and that can be detected. Either the presence, for example, in the flow cytometry, we can detect those kind of circulating tumor cells if they are present. In addition to that, circulating tumor DNA. So there are certain cells in the circulation which is representative of circulating cells. They also release certain molecules, including DNAs or proteins, so that can also be detected. DNA, RNA, and protein. In case of um, cancer patients, so control of tumor eradication, monitoring of tumor burden, choice of therapy. So these circulating tumor fragments can tell about the patient's status of disease in terms of their burden, in terms of their progression, And when they are, in terms of certain therapies, these can tell about, their progression, or can be used as a monitoring of those things. In healthy people, if these kind of tests are, applied, they can be classified in the early diagnosis. So, and similar to that, there are various specific type of assays which are known, which are used for identifying unknown primary cancers, single markers or integrative assays, and tumor specific marker, for example point mutation, gene derangement, copy number variation. So this is generally, various type of approaches which are used in terms of diagnosis, molecular diagnosis in oncology area. And so to summarize the overall take-home message with this today's, small brief presentation, is that the molecular diagnostics are increasingly used to 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 guide patient management, and it can be useful from diagnosis, starting from diagnosis to the final treatment, and also to monitor the disease progression or maybe the recovery of the patients. And it is very much useful in terms of cancer, infectious diseases, and congenital abnormalities, which includes various genetic disorders, either, in the prenatal condition, or, after birth. um, it provides the highest level of reliability, because, again, in terms of COVID also, you find, and, everybody prefers nowadays RT-PCR tests rather than going to the TrueNet or maybe, antigen antibody. So again, this is just one of the example of giving accurate, reliable, and robust results. And these molecular methods are applicable to research and clinics. And research wise, it is more useful in terms of, developing new mechanisms, sorry, identifying new mechanisms, and also to develop improved therapeutics in long term. So these methods are used in terms of, characterizing those proteins or identifying particular mutations, and then associated mechanisms. And eventually it is applicable to clinics in terms of, developing newer methods and developing new biomarkers. So with this, I think, um, I hope that I have given you some idea about what is molecular diagnostics. I'm sorry that most of the things I discussed, they are in terms of, more related to the clinical or application point of view. But that is what, it was decided that, and most of these things you will be, learning during your PhD or maybe your, curriculum. And the same course also, you will be learning about more detailed methodology or these techniques. So I hope that I may be able to communicate some of the part which maybe, you may be able to understand. And you are always welcome to discuss. And I would be happy if you have any question.